Welcome to our podcast on limb development. These are the topics that we'll be covering in this podcast: the anatomy of the developing limb, and we're really going to spend very little time talking about the anatomy and the bones and the cells and the tissues. We've really already constructed those in other sections of the course, but we're primarily going to focus on the formation of pattern. How do we determine three dimensions in this fairly symmetrical object that we begin with? That's going to require a discussion of cellular interactions, various inductive chemical signals, and then we're going to finish off talking about homeotic genes and how they function in the determination of pattern. So with limb development, much like everything else we've seen all semester long, we're going to begin in the fourth week of development. And when we look at the ventrolateral portion of the body wall, we can see in the more posterior position, we're going to have the leg bud. In a more anterior or cranial position, we're going to see the developing forelimb bud. We can call this the forelimb and this the hind limb. Of course, we're talking about chick embryonic development in this particular section. We use a lot of that. So we talk about the wing bud and the leg bud. Chick embryos are extremely useful models for development. One, you don't have to sacrifice a mother to obtain the fetuses. And because the eggs are laid freshly fertilized, you can very easily control the stage of development of the embryos to be used experimentally. Now, if we take one of the limb buds and cut it, this would almost be like a coronal section. And we're going to have three-dimensional sections through these, so you're going to have to stick with us as we go. But here we're looking at the dorsal surface in this histological slide. This is the ventral surface. In the background, if you will, coming at you would be the caudal portion of the embryo. The cranial portion is away from us. And what we're looking at here, this is going to be the distal growing tip of our limb bud. So this is going to be distal, closer to the body. This is going to be proximal. Those are the three dimensions that we're going to deal with when we talk about our developing limb. Now, the other way we can describe developing a limb essentially is an outer covering of skin. In this case, this is going to be our ectoderm. And the majority of our ectoderm that's covering it, once we get to this very distal zone and then going completely around the periphery like a belt around your waist, is going to be raised columnar type epithelial cells that we call the ectodermal ridge, and we'll see that in the next slide. And then at the very tip, this is going to be the most exaggerated of our ectodermal ridge, and since this is the most distal away, this is referred to as the apical ectodermal ridge, or we abbreviate that often AER. Now, this is going to be an important organizing and an abductive center, and we'll spend a lot of time looking at it and the materials and the chemicals that are produced. We also, when we look at our mesoderm, this is going to be undifferentiated somatic mesoderm and lateral plate mesoderm. We can divide this into a couple of different regions. First, we have the mesoderm that is immediately adjacent to the apical ectodermal ridge, and this is going to be the progress zone. And then we also have this posterior margin. Now, I liken it to the armpit region, sort of in the back of the limb. And this is going to be referred to as the zone of polarizing activity, or the ZPA, and that's how we abbreviate that. And we'll go into much more detail about all of these different components. But these are going to be the four players that we're going to be dealing with: the ectoderm, the ectodermal ridge, including the AER, then from the mesoderm portion, the ZPA, and then the progress zone. So in the scanning electron micrographic view, here we have the dorsal surface. Here's the ventral surface of our developing limb, proximal, close to the body wall here. This is the distal portion, and I think you can easily now see our ectodermal ridge, these raised cells, and at the very tip, the tallest of these cells is going to be along the apical ectodermal ridge, or the AER. This is just a light level view, again, showing the hind limb buds and the forelimb buds, these being wings and these being legs in our chick embryo. So now as we move through sort of the developmental stages that we're looking at, again, not really getting into the anatomy here. We started at 4th week. So now in week 5, we're going to see our limb buds are expanding at the distal ends. These are going to form what is referred to as the hand plate, and it's going to look more like a flipper or a paddle than necessarily it will hands and fingers or appendages at this time. Also, when we look at the bones, the ones that are closer to the body, the more proximal portion, this is going to be that single bone in our forelimb. We're talking about the humerus, and the hind limb, we're talking about our femur. We're going to see those mesodermal cells start to undergo this condensation event and chondrification. And this is going to occur the more proximal portions first, the more distal portions, this will occur a little bit later. Here we see an illustration of an embryo around week 5, and we can see that instead of a nice rounded limb bud, we have extended and expanded the distalmost portion into what we know as a hand plate, this paddle-like surface. Now, week 6, we're going to continue to extend our limb bud, but the hand plate is going to begin to distinguish itself. We are going to have chondrification centers that are going to occur in specific locations in the hand plate, and these are going to give rise to our digits. And so at this point, they're referred to as digital rays, and these are going to be the precursors to the phalanges in our forelimbs. Now, we can see in the illustration the lower or the hind limb, it lags a little bit further behind. This possibly has to do with the flow of blood, but certainly the forelimb is going to developmentally be very far ahead of what we see with the hind limb, especially at first. Now, a lot's going to happen week 7 and 8. We're going to begin to form joints. The limbs are going to rotate 90 degrees, so instead of being parallel, now they're going to have this more ventromedial type of positioning to them. We're going to see the digital rays continue to develop. We're going to undergo osteogenesis to form bone. And the separation of our individual digits is largely going to occur because of apoptosis of the mesodermal tissue in between our digits. So here we can see week 7, we're beginning to hollow out those positions with apoptosis between the digits, and by week 8, we have something that very much looks distinguishable as both a hand and our feet with distinct individual digits. Now, other components that we see, certainly cartilage, bone, ligaments, blood vessels. This is going to be derived from that limb mesenchyme, that mesodermal tissue, the somatic mesoderm and our lateral mesoderm. Limb muscles, this is going to be from our myotome. And then we also have a supply of neural crest cells. These are going to form the myelin sheaths around our peripheral nerves, both motor and sensory neurons that will innervate the limb. Now, we mentioned how the initial limb bud, it's rather an asymmetrical mass, and what we have to do is we have to convert this mass into a 3-dimensional structure. And in some ways, it's almost like 4-dimensional, because we're going to have, as we recall earlier, our proximal distal axis, anterior posterior, dorsal ventral. So armpit to fingernails, thumb to pinky, back of hand to palm. But then again, that 4th dimension, we've got to understand that we're a right hand versus a left hand. So there's almost that right-left positioning, but we can sort of infiltrate that in with the other 3. Here's our illustration relative to the chick limb, and we're going to continue to use this because this is the model that we're going to see for a lot of our experiments to help us decipher the different signals that are helping us shape this pattern. And even though it looks very different than we might think of as a human limb, we can see the single bone, in this case our humerus, radius and ulna. Then we have our carpals and our phalanges. And when we look at a chick limb, especially the wing, we have digit number 2, digit number 3, and digit number 4. And this is the pattern, the normal pattern that we're going to be working with and manipulating to understand these different chemical and molecular signals to help shape pattern. So the first thing we're going to look at is the role of the AER and the relationship it has to the progress zone. And remember, these are those mesodermal cells immediately underneath the apical ectodermal ridge. We see that both are essential for the outgrowth of the limb bud. Certainly the mesodermal cells are going to be the building blocks to extend the limb, but they are under the control of chemical signals from the apical ectodermal ridge that keep these cells in a proliferative state. Now, the signals that we see certainly fibroblast growth factors 8 and 4. These are going to be produced by the apical ectodermal ridge, and they are going to induce the cells of the progress zone to continue to stay in a state of mitotic activity. Again, as this happens, you're going to extend the limb. This is the growing portion of the limb. But as the cells, as the apical ectodermal ridge grows away from the tissue, those mesodermal cells are going to get further and further away from the chemical. They're going to be unable to diffuse, and finally you will fall below a threshold level, at which point the cells, the mesodermal cells, will no longer be under the mitotic influence of the apical ectodermal ridge, and they can follow a different specification and differentiation pathway to become cartilage or bone connective tissue. Now, this is an experiment where at differing days, the apical ectodermal ridge was removed from the developing embryo and the embryo allowed to develop to completion. So after 3 days, when you remove the apical ectodermal ridge, this is, remember, our normal pattern: humerus, radius, ulna, carpals, and then our 2 digits. We see that we get a truncated limb. If we wait 3.5 days of development, and to give you some frame of reference, chickens hatch after 21 days of gestation. So this is very early for a chick embryo. So when we look 3.5 days, we get a little bit more of our radius and ulna developed. And if we remove it after 4 days, we get the complete radius and ulna and be able to see some tarsals, but we don't finish the more distal structures. So this shows that this tissue, as we allow it to extend, more and more of these cells that have moved away from the mitotic influence of the apical ectodermal ridge are programmed to form the adult structures. Here's another experiment. We remove the apical ectodermal ridge, but we implant latex-coated beads that are soaked in FGF that will slowly, almost like a time-release capsule, release the materials. And at this point, when we remove the apical ectodermal ridge, we would predict that we would get a truncated limb, much like we see here. However, with the addition of the beads, we practically get a normal limb. Now, we can't argue that these distal portions are normal, but we can see humerus, radius, and ulna. We can see 1, 2, and 3, even though they are somewhat syndactylic, joined together. I hope you can appreciate how the addition of FGF was able to restore some of the pattern formation to these limbs. It didn't reproduce it exactly, but it was able to reproduce it in part. In the newest edition of this textbook, this was online and I was able to find this. It shows the apical ectodermal ridge in red, the FGFs that are produced that maintain these cells in the progress zone. But I hope you can appreciate from this that it's more than just the FGFs. Certainly FGF is going to play a big role in this, but here we have Indian hedgehog again. We have noggin, that BMP binding protein. We have our WNTs that we've mentioned already that are critical in limb development, the beta-catenins, and then transforming growth factors and the transforming growth factor receptors are all critical in this extension of the limb, the formation of where the joints are going to form, and the fibrous connective tissue that are going to be part of the joints. These are all critical in playing a role in that pattern formation. Now, we mentioned the relationship between AER and the progress zone. We're going to see that not only does the AER have an influence on the progress zone, but secretions from the progress zone are also critical to keep the AER in cycle. So both are positively feeding back to each other to keep the AER producing the FGFs and to keep those mesodermal cells in a proliferative state. They're sending signals back. We haven't completely deciphered all those signals, but we know they are essential and critical. Now, the other thing to mention is FGFs are also important for the specification of where the limb is going to form. And again, we do the experiment where we take a latex-coated bead And instead of putting it in place of a limb, we actually are going to transplant that on the side of the embryo where you shouldn't form a limb. Now, in this case, we're going to talk about the ZPA region, that posterior region, and this embryo was stained for sonic hedgehog, which is going to be characteristic of cells from the ZPA, and we can see it's posterior on all four of our normal limbs, but we also see that it is expressed on this induced ectopic limb that's forming, even though it's somewhat backward. It is going to be focalized to one area of the developing limb, which really speaks to the inductive effect of FGF on the formation of where a limb is going to occur. This is a hypothesis that really illustrates this idea that the further away the apical ectodermal ridge gets from the mesodermal cell, then the more we're going to develop proximal structures. And so this is not just distance, this is what they're referring to as time in the progress zone. So if we have cells that first are going to be lost from the influence of the AER, it's going to be under the influence for the shortest amount of time. And it's going to form the more proximal structure. Well, as the limb grows, we have the second set of cells that are under the influence for much longer before they leave. That's going to be, again, more time, a more distal structure, and we have, all along the way, we're going to have these cells. So there appears to be a relationship, and it's not necessarily going to be mathematical and have this nice linear pattern to it. But I hope you can appreciate the concept of time in the progress zone, which would also be equivalent to distance away from the apical ectodermal ridge, may be critical in dictating a proximal to distal pattern in the limb. So in a flow chart, we're going to see that we have the apical ectodermal ridge, we have the progress zone, and we can see the FGF signals establish the progress zone, maintain it. Signals that we're not completely aware of also help maintain the apical ectodermal ridge, and that's going to be critical for our proximo-distal pattern formation. Let's turn our attention now to the ZPA, zone of polarizing activity. We've already mentioned that it is in this posterior margin of the limbs, again, like I like to refer to as the armpit region. And this is thought to define the anterior-posterior axis. So this is going to define what is a thumb, what is a pinky. And we really have gained a lot of information from this because before the signals were even known about what's being produced there, scientists were able to dissect pieces of ZPA regions and explant them into additional places. So you basically would have ectopic ZPA activity in places that it normally didn't occur. And what we see is when you do that, you have malformations with the digits. And so we're going to expand on how the ZPA can control digits as well as this anterior-posterior axis. And we're going to see that much like the progress zone, the ZPA also is able to induce the apical ectodermal ridge by feedback mechanisms and maintain the AER in its state of being able to induce the progress zone. Now, we've talked about the FGFs and how they can induce the formation of a limb, how we keep the progress zone moving, and now we're talking about the AER producing something. Well, any substance that is produced that helps to control pattern, this being a concentration of a vitamin such as retinoic acid that we'll talk about, proteins such as sonic hedgehog, bone morphogenetic proteins, FGF, transforming growth factor beta, all of these we can refer to then as morphogens, morpho meaning form, and gen meaning generating. So certainly the signals produced by the ZPA, we can refer to them as morphogens. In this experiment, what we first examine is we have a normal limb. We can see the limb here. This is going to be anterior posterior, and here's our ZPA activity. And the hypothesis is that the tissue in the limb, when we look at the concentration of material produced, the concentration is going to be highest closer to the actual source. As you move further away toward the anterior portion, the concentration of the morphogen goes down to a point that it is below a threshold and you can't detect it. Well, it's believed then that that concentration effect determines digit identity. So we can see that in the highest concentration zone, we produce digit 4, moderate concentration zone, digit 3, and the lowest concentration zone, we produce digit 2. So we would predict if we could mimic that on both sides, and if this were in fact the case, and simply by transplanting a portion of a ZPA here, we should be able to get digit 4, digit 3, digit 2 in the opposite direction, and that's exactly what we see, a mirror image duplication of the digits. There we see 4, 3, and 2 mirror imaged what the normal endogenous digit identity would be. You can take that even further. You take a few cells, not a lot, but just a few cells, so you lower the concentration. You would expect possibly to get a digit 3 if you didn't go above that threshold. And by using varying amounts, you can control the digit that is produced. You can see we've only reproduced digit number 2. So there's something here with digit identity and the ZPA. So with this model, what we're looking at is we are looking at this morphogen that can diffuse across distances throughout the ZPA. And in that case, we've got evidence to correlate with a diffusible morphogen. But can we prove what it is? Can we substitute something that's going to mimic what's going on there? And really, that's difficult at best to do. But what we can say and know, especially from the illustration we saw earlier, is that sonic hedgehog, a very important inductive agent that we see throughout development, that is produced by the ZPA, and it could potentially be the source of this morphogenetic activity. One place we saw it was handedness in the embryo. Here we certainly have patterning, and so it's possible sonic hedgehog might be that smoking gun, if you will, of a potential morphogen helping to determine our digit identity. There's the image again, and we can see sonic hedgehog expressed in the ZPA of our 4 normal limbs, but it's also expressed in the portion of our induced ectopic limb. Now, as we continue to look for potentially morphogens in the ZPA, we can also see that in addition to sonic hedgehog, we can see that we have the transcription factor DHAND is produced. DHAND is also going to be expressed in that posterior marginal zone. DHAND controls the expression of sonic hedgehog. And once again, if we overexpress DHAND, we are going to cause the limb and the digits to duplicate, not necessarily in the same way we saw with sonic hedgehog, but apparently we do have a relationship between the transcription factor for sonic hedgehog and for duplication of digits. So as we continue to look for indications, we can add in extra material. Like the extra ZPA, we can add in beads that are soaked with various materials. But another thing that we can do is experiments where we can actually knock out genes, where we eliminate the genes and see what happens. In this particular case, and we'll look at this part first here, when we knock out sonic hedgehog in mice models, for instance, that we can still see that, yes, we do have limbs begin to form. Remember, that was under the effect of FGF. But we don't get those distal limb structures. They fail to form. Digits don't form correctly. And we can further support that in a mouse model that's called the extra toes model. And again, polydactyly is going to be that terminology used when you have extra digits. And here we have an individual with polydactyly. At first it looks, fairly normal. We can see a thumb, we can see a pinky, index finger, your fourth finger. But wow, here we have this duplication of the middle digit. And so that is our polydactyly. We can see the fusion of that toward the base. But when we see these extra digits, we also correlate that with an increased anterior expression of sonic hedgehog. So we're building a case. It's like a lawyer getting the evidence all together. We can't reproduce the crime yet, but we're getting closer and closer to having this evidence available to show, you know what, it really does look like this is the culprit. But not to leave anyone else out, we have bone morphogenetic protein is also expressed in the ZPA, and we know BMP is influenced by sonic hedgehog as well. And so the current model that we're sort of putting together now really looks like that bone morphogenetic protein is what may actually determine digit identity. So is it a 2? Is it a 3? Is it a 4? Sonic hedgehog, however, appears to be that is determining digit number. And again, we could go through a lot of experimental protocols and papers to try to really bring this point home, but this is sort of a synopsis of what we know as of right now, the players in the forming of the pattern of the limb. One of the earliest morphogens that we looked at and really began to understand its role in pattern formation is retinoic acid. Now, retinoic acid is in the ZPA, and we do see that it can induce the formation of extra digits. But if we transplant a bead soaked with retinoic acid into that posterior zone, into the anterior zone, we see that it induces the cells around it to form a new ZPA to begin to produce sonic hedgehog, and that does lead to extra digits. So retinoic acid, we can see that it is going to be involved in the induction of cells that recreate sonic hedgehog, and therefore it is going to be that initiates the formation of the ZPA. Now, retinoic acid, much like FGF in this case, it may also be able to initiate the formation of a limb, certainly as we talk about forming the ZPA. But what we also see is that if we block retinoic acid, and specifically we prevent retinoic acid from binding to its receptor. So if we block retinoic acid, we see that we are going to prevent the outgrowth of the limb bud. So again, here's some evidence that we don't know exactly what to do with it. Retinoic acid is playing a role. We haven't put together all the pieces yet. Retinoic acid binding proteins and retinoic acid receptors, we're just getting a handle on those, but retinoic acid may be one of the players of many helping to induce 3D pattern formation in our developing embryo. So here we add to our flow chart. We have the AER in the progress zone. Now we've got our ZPA. And ZPA, certainly, we are going to form sonic hedgehog and BMP signaling that is going to potentially have an impact on the progress zone for digit identity and digit number. And certainly our anterior-posterior axis is going to be determined through that as well. The ZPA is going to be signaling the apical ectodermal ridge. And it's possible that FGF is going to lead to the formation of the polarizing region and therefore be critical for initiating the formation of a limb. So it may be FGF turns on retinoic acid, that turns on DHAND, that turns on sonic hedgehog, that functions along with BMP. There's going to be a signaling cascade that someday hopefully we'll be able to decipher.